speakers for this wonderful conferences. I'll talk about multiple systems in the dopamine systems today. So let's start. So first, please imagine something threatening to you. Now, I have a question for you. Have you experienced the outcome of the threats? I guess most of you never experience the ultimate outcomes of these threats. Now, please imagine something rewarding to you. And then, I'd like to ask you the same question now. Have you ever experienced the reward? I guess, in most of the case, yes. So apparently, there is a critical asymmetry in these two types of knowledge. On one hand, you experience and then learn in a daily life. But for the other, you seldom experience but really want to predict and avoid beforehand. So how does brain handle this asymmetry for learning? So information for both reward and threat is important for survival. And our understanding of learning of these has been advanced in different fields: reinforcement learning and fear conditioning. So today, I'd like to discuss how animals evaluate reward and threat in a framework of reinforcement learning. So as you already know, so most of you know, reinforcement learning theory has developed together with studies of machine learning and animal behavior, and also studies of dopamine. So it is widely accepted that dopamine conveys information about reward. So dopamine neurons are excited when reward was delivered unexpectedly, but do not respond when the reward is delivered as expected, and shows dip or inhibition when reward is omitted. So Schutz, Dayan, Montague, and Sejonosky formalize this activity pattern of dopamine as reward prediction error, or RPE, discrepancy of actual and predicted value of reward. So importantly, the reward prediction error is bidirectional. So excitation with outcomes better than predicted, and inhibition with outcome worse than predicted. And similar patterns have been seen when measuring dopamine concentration in the striatum. So using a lottery task in rats, Hart and Philip show that dopamine concentration increases more here with unexpected win, and decreases more with unexpected loss, consistent with bidirectional RPE. According to learning theories, bidirectional reward prediction error may function as a teaching signal to reinforce actions to maximize reward and minimize punishment in the future. So far, I only talked about reinforcement learning with dopamine neurons related to reward, so related to gain or loss of reward. On the other hand, there had been a lot of controversy about dopamine function related to aversive stimuli. So because inhibition of dopamine activity might not be enough to signal aversive stimuli, and because there are several structural asymmetry in reward and punishment learning, so many groups, including Buho and Dayan, propose that dopamine represents reward, whereas another molecule, such as serotonin, may represent punishment. Later, Fiorio reported his observation that dopamine neurons did not show significant inhibition with aversive stimulus compared to neutral stimulus, concluding that dopamine responses, dopamine represents reward only, supporting these ideas. Although his data was contradicted by many other groups. So one of the reasons for the contradiction is that So this and other studies relied on indirect method to identify dopamine neurons. So Cohen and Hessler established optogenetic method to unambiguously identify dopamine neurons. We tested whether optogenetically identified dopamine neurons really ignore negative values of aversive stimuli. So throughout the training, we used very low probability of reward because unnaturally high probability of reward during training causes generalization of reward cue, causing small excitation of dopamine neurons with all the cues. So under low probability of reward, we observed that at the single neuron level, many dopamine neurons are indeed excited with reward predicting cue and inhibited by an aversive ABAF predicting cue. Moreover, if a cue, this cue, predicted reward and ABAF in a probabilistic manner, dopamine neurons showed intermediate activity to that cue. The results indicate that dopamine neurons monotonically represent combined value of both valences, appetitive and aversive. Taken together, dopamine signals can be used as common currency representing prediction error of value. In this way, theoretically, bidirectional RPE can be used as teaching signals to support both learning of reward and learning from punishment. However, not all the dopamine activity can be easily explained by RPE. For example, dopamine neurons are activated by non-rewarding stimuli, such as novel tone or unexpected door opening. Kakade and Diane interpreted novelty responses of dopamine neurons as a novelty bonus in a framework of RPE because maybe novelty itself is rewarding or because a novel stimulus predicts potential reward, although the exact function was not known. Further, multiple studies, including ours, also found that at least some dopamine neurons are excited by aversive stimulus. The excitation by punishment violates the learning model because animals presumably do not want to reinforce actions yielding punishment. So there are several models to explain these observations too. For example, two-factor theory is a popular idea for learning of active avoidance. In this theory, an animal first learns negative value of punishment by inhibition of dopamine neurons by punishment. At the next step, an animal happens to avoid the punishment and dopamine neurons are excited, in this case, excited with unexpected no punishment or termination of punishment, reinforcing the avoidance behavior. Eventually, the punishment predicting cue will become safety predicting cue, which may also cause excitation of dopamine neurons. Thus, some results for excitation of dopamine neurons by aversive stimuli may come from positive values of safety. In these ways, on one hand, accumulating evidences indicate an unusual dopamine activity that may violate reward prediction error theory, and on the other hand, theories explain those activities within a model of reward prediction error. However, before arguing which is correct, we need to consider diversity of dopamine neurons. But I also have to point out that some of the studies of diversity of dopamine neurons are also misleading. So we need to categorize the diverse dopamine neurons to determine which features in diversity are variation in a single type of dopamine neurons and which features determine fundamental distinction of function. So in order to tackle this controversial question, understanding the anatomical basis of the dopamine striatum system is a key step. So in these photos, dopamine axons are genetically labeled with a red fluorescent marker. So you can see the striatum, So these are striatum and cortex and thalamus and hypothalamus. This is a mouse brain. So as you see, the striatum is the predominant receiver of dopamine projection compared to other brain areas. The striatum resides beneath the cortex and receives projection from most cortical areas. Then, the striatum indirectly sends outputs downstream to the brain stem and also sends information back to the cortex through the thalamus. From these anatomical features, the striatum is thought to gather and filter information from cortex with dopamine modulating information flow through the striatum. Because there is topographical relation between cortex and cortical areas and striatal areas, the important question is whether dopamine neurons broadcast reward-related teaching signals throughout the striatum, or there are unique striatal areas that receive qualitatively different information. Anatomically, single dopamine neurons tend to project to specific brain areas. We reason that if some dopamine neurons show different activity from canonical dopamine neurons, those dopamine neurons should receive different information from presynaptic neurons. So if this is the case, we might be able to detect the difference anatomically. We use modified rabies virus to map location of presynaptic neurons to dopamine neurons. So different from conventional tracing, this method allows retrograde tracing from a specific cell type. Using this method, we mapped inputs to dopamine neurons in the whole brain. So we found that dopamine neurons in VTA and SNC receive inputs from largely overlapped but slightly different input areas, brain areas. Since this study did not distinguish projection targets of dopamine neurons, we did not know exactly which dopamine subpopulation contributed the observed differences. So next, we examined inputs to dopamine neurons with different projection targets. We compared distribution of monosynaptic inputs to dopamine subpopulations with 8 different projection targets. So these are the projection targets of dopamine neurons, and green dots are presynaptic neurons to those subpopulation of dopamine neurons. So surprisingly, we found that most dopamine neurons received a very similar set of inputs, except that dopamine neurons that project to the posterior tail of the striatum showed different input pattern. I will call this area tail of the striatum TS in the rest of my talk. We quantified the number of inputs from each brain area. While the ventral patch, in the ventral striatum provides abundant inputs to other dopamine subpopulations, TS projecting dopamine neurons receive much fewer inputs from this area, but instead receive many inputs from unique areas such as globus pallidus and zona insata. So as you see here, there are more or less difference in input patterns between each dopamine subpopulation. So if you compare only one by one, you can always find some difference. In order to categorize dopamine subpopulations, we compared similarity of input patterns. We examined the pairwise correlation of patterns of inputs to each subpopulation of dopamine neurons and confirmed that TS projecting dopamine neurons were outliers among 8 subpopulations of dopamine neurons. This screening step suggested that TS projecting dopamine neurons may have a unique function. Next, we examined the activity pattern of these dopamine neurons. We recorded population activity of dopamine axons using fiber fluorometry, or people often call fiber photometry. While mice performed classical conditioning tasks, we found that dopamine axons in TS are uniquely activated by aversive air puff or air puff predicting cue. If we compare responses to air path, the excitation was localized in TS, the most posterior part of the striatum. Our results show that TS projecting dopamine neurons are unique not only in connectivity but also in activity. We next examined the activity of TS projecting dopamine neurons more carefully with the ventral striatum projecting dopamine neurons as a comparison. Dopamine axons in the ventral striatum are strongly activated by water reward, and their activity monotonically correlates with water amounts, consistent with the idea of value representation. On the other hand, dopamine axons activity in TS did not correlate with reward value. Instead, the activity of dopamine axons in TS correlates with intensity of tone and air path. Because these stimuli at high intensities are considered aversive, we next examined responses to other types of aversive stimuli. Dopamine axons in VS were inhibited by all types of aversive stimuli we tested. Here, air puff, beta water, and omission of expected water, consistent with the negative values of these stimuli. However, dopamine axons in TS were activated by only subset of aversive stimuli. Here, only by air puff, but not by beta water or water omission. In addition, dopamine axons in TS were strongly excited by novel stimuli, whereas dopamine axons in VS did not respond to novel stimuli. We observed similar activity when we recorded from the cell body of TS projecting dopamine neurons using rabies virus. Thus, these differences are not caused by modification at the level of axons, but the activity of dopamine neurons are different. In short, TS projecting dopamine neurons respond to air path, tone, light, or odor, but not to beta water, reward omission, or reward. So it seems that TS projecting dopamine neurons do not encode valence, but respond to various external stimuli in the environment, especially high-intensity stimuli or novel stimuli. So what is the function of TS projecting dopamine neurons? Anatomically, recent studies divided the striatum into sub-areas according to cortical inputs and show that TS receives cortical projection from sensory areas such as visual and auditory cortices and temporal association cortex. This suggests that the dopamine signals of high intensity or novelty may directly impact sensory information flow. In summary, although excitation to non-rewarding stimuli was often discussed as evidences against dopamine RPE theory, dopamine responses to non-rewarding stimuli do not coincide with value signals. In other words, RPE is not contaminated, at least in some brain areas, strongly suggesting a role of dopamine in value-based evaluation in these areas. On the other hand, dopamine responses to novelty and high-intensity stimuli are localized in TS, suggesting special function in sensory information flow. So what is the exact function of dopamine in TS? We first optogenetically activated dopamine axons using channel rhodopsin. Mice performed choice tasks. They poke a center port to initiate the trial and chose left or right water port depending on the outcome. One port had outcome of X in addition to water reward. With this task, mice preferred the port with extra water and avoided the port with air puff or beta water. Next, we activated dopamine axons in the ventral striatum. Consistent with rewarding or reinforcing effects of dopamine, mice preferred optogenetic activation of dopamine axons in the ventral striatum. In contrast, mice avoided optogenetic activation of dopamine axons in TS, suggesting qualitatively different function between these subpopulations. So next, we ablated dopamine neurons using 6-OHDA, a toxin that specifically kills dopamine neurons. So we specifically ablated TS projecting dopamine neurons by injecting the toxin in TS, and then tested their choice behaviors. As I mentioned, normal mice learn to avoid the port with air puff. However, after ablation of TS projecting dopamine neurons, individual mice kept their own choice bias. And as a population, ablation mice did not show tendency to avoid an air puff port. In contrast, ablation mice were able to learn to avoid port associated with bitter water or less amount of water. These results show that dopamine in TS plays a critical role in learning from punishment, but not from all punishment. The results indicate two important points. First, different brain systems are responsible for learning from different types of punishment. So if we study punishment, we should distinguish different types of punishment. Second, dopamine in TS is specialized for learning to avoid external threatening stimuli, consistent with our observation of activity pattern of TS projecting dopamine neurons. By the way, the other punishment could be categorized into loss of reward, which may solely engage canonical dopamine neurons of value system. So how could we explain the function of dopamine in TS compared to canonical dopamine in reinforcement learning? As I showed at the beginning, dopamine RPE can enhance reward prediction and also reinforce rewarding action. What about the signals from TS projecting dopamine neurons? We noticed that although their functions are qualitatively different, there are some similarities in these systems. As for activity, TS projecting dopamine neurons showed components of prediction error, such as excitation by threat predicting cue and reduction of responses with expectation. As for function, optogenetic activation or ablation of TS projecting dopamine neurons did not affect initial responses, but affected subsequent trials, suggesting reinforcing effects. Then, TS projecting dopamine neurons may function in a similar algorithm as canonical dopamine neurons, namely by Pavlovian prediction. So dopamine neurons may send threat prediction error signals to TS to enhance threat prediction, facilitating downstream action according to threat prediction. Thus, we propose that there are two axes of teaching signals by distinct dopamine neurons. In the real life, animals usually need to consider both reward and potential threat. So if value prediction system and threat prediction system are working in parallel, alteration of the balance between these systems might change the behavioral choice. We are currently examining this possibility in several different behavioral paradigms in more naturalistic settings. So here, this is one example. So here, we gave a new toy to mice in a familiar environment and observed their behavior while they freely explored. When animals encounter a novel object, they show characteristic behaviors: cautious approach and quick read. So slowly approach and quick read. So MOSIC is a method to classify behaviors by detecting mouse body shapes like this using depth camera. By behavioral classification with MOSIC, we could capture approach and retreat syllables unique to novelty encounter. So approach avoidance conflicts to novelty such as this are observed across various animals, including human beings. William James, a famous American psychologist, was observing an alligator who tried to approach a human being. Then he wrote, so he was very calm, he wrote, "Since any entirely unknown object may be fraught with will or woe, nature implants contrary impulses to act on many classes of things. Thus, curiosity and timidity seem to shoot over into each other as quickly and to remain in as unstable equilibrium. Curiosity and fear form a couple of antagonistic emotions liable to be awakened by the same outward thing and manifestly both useful to the possessor. Dopamine in TS may regulate this balance. To study this possibility, we examine effects of ablation of TS projecting dopamine neurons on novel object exploration. As we have seen with mosaic, normal mice showed cautious approach retreat behaviors. Ablation of TS projecting dopamine neurons did not change this behavior. At the beginning. 2 minutes later, normal mice still show short bouts of approach-retreat behaviors. In stark contrast, TS dopamine ablation mice started to lose timidity toward the novel toy much faster than normal mice. These are raw traces of example mice. Control mice kept short bouts of approach-retreat behaviors for 5 minutes, but ablation mice lost these behaviors. These results suggest that two axes of dopamine systems play roles in event-by-event updating in more situations than we previously thought. If one system is suppressed, such as in our ablation mice, only one system, in this case, value system, predominantly enhances reward prediction, causing premature approach before enough investigation. In this way, we could interpret the unstable equilibrium during the updating period as risk assessment. Historically, it has been postulated that many psychiatric conditions are caused by abnormal absolute amount of dopamine, too much dopamine or too little dopamine. However, our results suggest that balance between multiple dopamine systems is important for normal risk assessment behaviors. So today, I first show that having bidirectional value axis is useful for animal learning. And then I describe that there may be two axes of dopamine systems. So why do animals use multiple axes of evaluation systems for learning? Why different types of punishment engage a different system? So I think that multiple axes give much flexibility to allow brain to fine-tune the learning algorithm separately. For example, while the value system does not respond to a novel stimulus until animals learn to associate the stimulus with reward, the threat system responds to novel stimulus strongly. So in other words, threat prediction is high for novel stimuli and maintained high until animals confirm safety. So thus dopamine systems are taking a cautious strategy, initialize high for threat and 0 for value. Further, while dopamine LP is bidirectional, dopamine threat prediction error does not signal threat omission, suggesting that omission of threat does not immediately erase threat prediction, which also makes sense for animal survival. In this way, we can interpret difference in activity rules in two dopamine systems as critical modification to learning algorithm to achieve each goal, while they might share similar mechanism of Pavlovian prediction as a working principle. With separate evaluation systems, dopamine may support animals' rich behavioral repertoire, which may not be possible with single axis. So in this sense, multiple axes of dopamine evaluation systems might be also related to ideas of multiple axes of emotion systems. There are different opinions about how to categorize emotions. So many models used to use valence and arousal axes, and more recent studies propose more axes or sometimes use valence and intensity axes. So it is difficult to determine which one is true because emotional studies are dealing with subjective internal states. Interestingly, Saskin and Anderson proposed that the facial expression may have functional meanings. So although both fear and disgust express negative values, the facial expression look like functionally opposite. So fear faces open eyes and nasal cavity widely as if they try to sense as many information as possible, whereas disgust faces try to minimize exposure to any stimulus. It is fascinating if combination of multiple dopamine systems could explain behavioral and facial outputs owing to various internal states, because then such objective measurements can advance understanding of normal and psychiatric conditions. So in this talk, I show that there are two axes of teaching signals by distinct dopamine neurons. And I'd like to emphasize that balance between these systems is important. So where these systems interact, whether they interact at the level of the brainstem or at higher cortical center via basal ganglia loops, is an important question for future. Okay, so I'd like to thank all of my lab members, especially William Menegas, who did all the works with TS, anatomy and activity and function, and also Hide Matsumoto, who is now working in Osaka, Osaka City University, with very, very patient single unit electrophysiologist. Thank you for attention.